welcome everybody to the plenary for the second day of the Biodiversity Genomics Conference. My name is Richard Durbin from the University of Cambridge, and it's my pleasure to chair this session and this talk. So, and the talk will be given by Adam Phillippy from NHGRI, and the subject, the title is Finishing the Complete Sequence of a Human Genome. So there are two things to introduce the speaker and the topic. Adam is in the intramural branch of NHGRI, the NIH's Genome Institute, where he heads the Genome Informatics section. And I just learned by a bit of Googling and talking to Adam before the talk that he actually left sort of his academic training for 4 years after his first degree in computer science and worked at the Institute for Genome Research, TIGER, in the early 2000s, which introduced him to genome assembly in practice. And then he went and did a PhD with Stephen Saltzberg, I presume with a clear picture of what he wanted to do. And then after he finished that in 2010, he set up his own group. And since then, he's really been at the forefront of genome assembly. And from my perspective, he always seems to aim to achieve perfection while maintaining practicality. And it's bringing those two things together that's the sort of ultimate, slightly quixotic goal, which actually I think we now are pretty close to achieving. And that's in large part due to Adam. So, I mean, this is a period around that time, 2010, which some people thought that assembly was either done or was boring. But now we can see it as really the Renaissance period for genome sequencing and possibly the start of a golden age. And Adam, I think, realized this very early and saw the potential of long reads. He built, and his group built, one of the most flexible assemblers, Canoe. He also saw the need to separate out the haplotypes, and they introduced trio-based assembly. And he built a group and became a key contributor and leader in many, many collaborative projects. And I've met him in the context of the Genome 10K assemblathons and then the Vertebrate Genomes Project, the VGP, where he has very impressively led the assembly group. And that's been a real joy to be part of over the last few years. And I've learned so much from that whole enterprise. So, and furthermore, he seems to be at the heart of not just that, but many of the other most challenging large-scale assembly efforts that are going on today. And that brings us to today's talk. We've heard many rounds of claims of completing the human genome sequence. I mean, in fact, I've been present at least two of those, starting with the famous White House Downing Street announcement in 2000, 20 years ago. And we all knew then that it wasn't really finished. It was just a sort of something that was represented everywhere. But there was a second completion, in quotes, in 2004. And that also wasn't far from being finished in reality. We know that genomes really are finite, they can be completed, and that's where we've got to. And it's really, in the last few years, Adam with Karen Meager have put together a consortium he's going to tell you about that have pretty much got an end-to-end complete sequence of the whole human genome. And so for me, it's a very special talk. It's a true milestone in genomics that we're about to hear about. It's the culmination, arguably, of 30 years of genome projects and the human genome sequencing, and it's absolutely the right person to be giving it. And so we're now going to hear the talk, which was pre-recorded, and following that, there'll be questions.
Hello, everyone. My name is Adam Phillippy. I'm a senior investigator at the US National Human Genome Research Institute, and I'm excited to share with you today some of the recent progress, very recent progress from the Telomere to Telomere Consortium, which I co-chair with Karen Meager and Evan Eichler. I would much rather be with you in person. Obviously, this talk is being pre-recorded, but to give you the feel of a live talk, I was working on my slides until the last minute. We'll do one take, and I've prepared very little. So hopefully you'll enjoy it. Let's jump in. This is a bit of a special year. We're 2 decades since the initial release of the Human Reference Genome Draft, which was announced on June 26, 2000 and published shortly thereafter in 2001. Obviously, for those that were around, we remember the competition between the Public Human Genome Project and the private Celera project, and both of those papers were published announcing the initial sequencing and finishing of the human genome. But there's a few caveats with those initial releases. The first is that it wasn't a complete genome. The genome had not been finished and it remains unfinished 20 years later. And they often talked about the human genome reference. We since talk more about a pan-genome representation of humans, and we're only sequencing one genome at a time. They were no different. So the goal of the telomere to telomere consortium was to complete these initial projects for at least one human genome. And so the title of the talk today is the complete sequence of a human genome, and that's been the goal of the T2T for the past few years. I think this is pretty well known within this community, but for those of you that are coming from conservation genomics or other areas, it's sometimes overlooked that the human genome reference is not as good as it's often advertised. And that misrepresentation even applies to my own institute. Genome.gov, NHGRI's homepage, has this quote on it still today that says, in the April 2003 version of the human reference, there are less than 400 gaps and 99% of the genome is finished. And that's not entirely accurate. What's being left out is the heterochromatin. The 99% figure is talking only about the euchromatic portion of the genome, and there's maybe 5 to 10% of the genome that goes forgotten if you only look at the euchromatin. And just because it's forgotten doesn't mean that it's not important. These are hugely consequential regions of the genome that include the centromeres, which are obviously critical for cell division, the telomeres, segmentally duplicated genes, which are often very quick in evolutionary terms when you look compared to our primate relatives. There's tandem gene arrays, especially on the sex chromosomes, ampliconic gene arrays, and there's an unknown number of errors and omissions in the current human reference that we're looking to correct and fill in. So if you tally it up, there's maybe 5 to 10% of the human genome that's really unmapped by current technologies. And if you look on this ideogram here to the right, there's a large human satellite array on chromosome 1, for instance, on chromosome 9, the short arms of the acrocentric chromosomes here, 13, 14, 15, 21, and 22, and a lot of heterochromatic sequence on the human Y. The ideogram displays them here in blue, I guess for two reasons. One is that they stain a little bit differently when you look at a typical banding pattern, but also they're completely absent from the current human reference. If you bring up GRCH38, the most recent human reference, it will be represented by giant stretches of ends for millions and millions of bases, also including the pink here, all of the human centromeres. To date, with the exception of the Y, have been completely unfinished. So why does this matter? I mentioned some of the scientific and functional reasons why this sequence is important, but there's also some less fully appreciated reasons. First, if you're missing reference sequence and you're doing a resequencing study of additional humans and you're trying to map to that incomplete reference, that causes a number of biases. So if you have sequencing reads that were sampled from any of these regions of the genome that have no reference sequence, and then you remap them, they will often mismap to the incorrect region of the genome, which causes artifacts in the form of maybe false positive SNP variants. Furthermore, because we don't know these regions, the variation within them is unexplored. So any of the linkage-based studies, GWAS studies that are being performed will not be able to link a lot of diseases to those regions if there's functional consequences. Third, if you're trying to do a functional study, understand the function of the centromere, understand the function of the rDNA arrays. If you don't have a reference sequence, it's very hard to study that. You can't synthesize constructs, you can't design probes. It really limits your ability to probe that sequence. And then lastly, we don't know what we don't know. Since these are really unexplored regions, we don't even know if we're missing potential genes and transcripts within these regions. So why has it taken so long? The short answer is repeats. Genomic repeats are the bane of genome assembly and even cloning in some cases, and that has really hampered our ability to probe into these regions over the past 2 decades. If we go back another 20 years, 40 years since we've been working on the genome assembly problem, and we've known that repeats are the primary barrier to genome sequencing and assembly since 1979. If you look at this first publication of a genome assembler by Roger Staden over 40 years ago. It has this key quote that the overlap of two of your gel readings need to be sufficiently long to distinguish them from being a repeat. And if so, you can then paste them together to make a more continuous sequence. I also love this other quote from the same paper. With modern fast sequencing techniques and suitable computer programs, it is now possible to sequence whole genomes without the need of restriction maps. It would almost work as the title of my paper, of my presentation today. But instead of talking about Nanopore and PacBio, the citations here are Sanger and Maxim Gilbert sequencing. And so 40 years ago, Roger Staden and Fred Sanger laying the groundwork for what we're going to talk about today. And that's really a new era of sequencing that has been introduced with single molecule long read sequencing. And the first technology that really made me start to believe that we could finish these remaining gaps in the human reference genome was the advent of ultra-long nanopore sequencing. And we published a paper as part of a consortium 2 years ago, where we introduced a protocol to do ultra-long read sequencing. We demonstrated that you could now sample reads over a megabase pair in length from a single strand, and that you could use that information to assemble very high quality genomes. Glenys Logsdon and the T2T consortium has been doing some great work on this recently to improve those protocols and apply them to our work. Shown here in the bottom left is our collaborator, Matt Luce, also showing the portability of nanopore sequencing and sequencing DNA in a hotel room at AGBT a few years back on a MinION device. But aside from the portability, it's really the length of the molecules that you can sequence that makes Nanopore very attractive for whole genome sequencing and assembly, because just like a jigsaw puzzle, when your pieces are much bigger, it's much easier to put the puzzle back together again. So with that technology in hand, my good friend and collaborator, Karen Miga, who co-founded the T2T project with me about 3 years ago, called me up. We had a video chat, and she expressed to me her strong desire to finish the human genome. I said, what a great coincidence. That's one of the aims of my section at NHGRI. When I joined NHGRI in 2015, one of the specific aims of my research was to finish these remaining gaps in the human genome. And I had been working on genome assembly methods for many years, specifically designed for PacBio nanopore sequencing. Karen is a satellite DNA expert, and so she had the knowledge and the expertise of these unfinished regions. And our two fields of expertise came together, complemented each other well to take on this project. And so we founded what we're calling the Telomere to Telomere Consortium. It's an open consortium, a community-based effort to generate the first complete assembly of a human genome. And we did a lot of thinking up front as to what would be the best genome to target. And we made a very pragmatic decision and selected what's called a complete hydatidiform mole cell line. These cell lines are originated from what's called a molar pregnancy, where a sperm fertilizes an egg that's lost its genome, and the sperm spontaneously duplicates its own genome. In the best case, you end up with a normal karyotype where it's fully homozygous. So each copy of the chromosomes is identical. In the case of CHM13, it had a normal 46XX cell line. So unfortunately, no Y. Those are essentially non-existent in these mole cell lines. But it fit the bill. It looked stable under karyotype. It had very little heterozygosity across the genome, and we could culture it up and get lots and lots of DNA to feed the sequencers. So starting with that CHM13 resource, we basically threw the kitchen sink as the saying goes, and sequenced now a total of 120x Nanopore ultra-long, 70x PacBio continuous long reads, 30x PacBio HiFi, 50x 10x genomics, 100x PCR-free Illumina, 35x Arima Hi-C, a BioNano optical map, and PacBio IsoSeq. And we publicly released all of this data. You can go to this link. It's all up on Amazon Web Services. You can download it for free without egress charge, play around with it, develop new methods. And that was really our intent in releasing this data set, that it would encourage development of new methods that would enable us to finally finish these remaining gaps in CHM13. My group did that as well. And we developed what's called the Canoe assembler, which works well for both Nanopore and PacBio data types. And I like to give a feeling of what the genome looked like 20 years ago versus what it looks like today with these modern technologies and algorithms like Canoe. And so here's an ideogram showing the state of the human genome in 2001 when it was announced finished to great fanfare. In today's dollars, this is over a $5 billion project. It was announced at the White House in the summer of 2000. And the ideogram here is showing the continuity of the assembly. So this isn't a typical banding diagram that you're used to seeing. Here, each of the bands is a contig, whether they're gray or black. I switch the color anytime there's a gap between two contigs. And so anywhere you see a gray or black horizontal band, that's one continuous piece of sequence. And the white are the regions that are still missing from the reference, so we have nothing to map them to. The takeaway from this figure is that even though the genome was announced finished, essentially in 2001, 2004, it was still heavily fragmented. And the average contig size in this assembly is about half a megabase pair, and hundreds to thousands of contigs per chromosome. There was many, many gaps. It was nowhere near a complete and finished genome. When we take the T2T data for CHM13 and run it through our Canoe assembly program, this is what the genome looks like as of last year. And so this is actually an assembly of just the Nanopore data that was collected on a Oxford Nanopore GridION instrument over the course of a summer a few years ago. Run through Canoe, this is the same banding diagram as I was showing before, where now you can see chromosomes like X are assembled from telomere to telomere in a single continuous assembly, unbroken with no gaps. Other chromosomes like 3, for instance, 11 are present in the assembly with maybe only 1 or 2 gaps. And this is an automated assembly, no human intervention, one set of sequencing reads, one person running the machine, one person running the assembly, compared to 20 years ago. And the results are just an order of magnitude improvement. I like to show that as just a testament to what the investment has been in genomics and sequencing technologies over the last 20 years, and the huge return that we've had on that investment and our ability to now sequence whole genomes at a resolution and completeness that was not even dreamable 20 years ago. And so this enabled us now to have, for instance, the first telomere to telomere assembly of a complete human X chromosome as published by our consortium this year for the human X chromosome. And the strategy that we used to get these first complete human chromosomes was really a Nanopore first to build a backbone, followed by PacBio polishing. And the idea for this approach is to really build off of the enormous lengths of the enormous size of reads that you can get with Nanopore sequencing. And in the case of the X centromere here, DXZ1 satellite array, it's 3.1 megabases of sequence. And comprising that array is a relatively small repeat unit of a few thousand base pairs that's duplicated many, many times. Each of the little vertical gray bars, there's so many here that they almost bleed together, is a canonical copy of the repeat. And the little colored vertical bars are unique variants that exist within the array, where there's a copy of the repeat that has a variant different from the canonical version. And in the assembled form at the very bottom, you can see the layout of those unique variants relative to the finished structure. And the one highlighted here with a little caret is actually a line element inserted into the array. And what I'm showing you here with these 12 reads is an example of tiling across that 3 megabase pair satellite array with just 12 ultra long Nanopore reads. So the reads are so long that you can get across this array, anchoring the reads together with these, what we would call signpost or landmarks, to orient yourself in space as you tile across that array using just 12 of these ultra long reads. Karen Miga, when she first constructed this array, used this technique. She first identified what the unique repeat variants were using a combination of PacBio sequencing and Illumina K-mers and so forth, identified those unique landmarks in the Nanopore reads, and then stitched them together using those signposts to guide her way across the centromere. That gave us a backbone, but there was still a lower sequencing quality in the consensus sequence because it was entirely based on Nanopore. The best consensus quality we could get at the time was maybe 99%, maybe a little bit better. And so there was still a number of errors within the final assembly. And that's illustrated with this plot here on the bottom left, which is coming from Mitchell Volger in Evan Eichler's lab. When he took PacBio data and aligned it to our assembled backbone, we noticed that the mapping was non-uniform, which is not something we would expect. Shown here in black is the depth of coverage of the primary mapped allele, and in red is the depth of coverage of the secondary mapped allele. Because this is a homozygous cell line, we expect no secondary allele coverage, and we expect high and uniform primary allele coverage. But what we saw was a peak in some regions of almost double coverage of the primary allele, and a deficit in other places where there was no mapped coverage of the primary allele. And this was the first flag to us that there was something slightly wrong with our initial assembly. Even though we were very confident in the structure of the assembly because it was guided by these unique variants, something was wrong with the consensus. So we developed a number of polishing approaches that would take PacBio reads and uniquely anchor them to our Nanopore backbone and run what we call polishing, where we confidently map the reads to that backbone, run that PacBio data in this case through a consensus algorithm to correct any errors in the initial assembly. When we revalidate at the end of the process, we now get uniform primary allele coverage, very low secondary allele coverage, which is what we expect. And this is the assembly that was released in the paper this year, and is the assembly that we're confident is correct because now it's been validated by a number of orthogonal means. That led to the complete X chromosome. This is the first human chromosome to be completed telomere to telomere through the centromere without gaps. And just to give you an idea of the types of regions of the genome that present problems for these approaches, there's a gauge tandem gene family, which spans about 190 kilobases. There's the DXZ1 satellite array at the centromere, which I was just talking about. There's a number of segmental duplications within the X and to other regions in the genome that are over 100 kilobases in length. And there's another satellite array, DXZ4, which is shorter, but also presents problems for assembly at 165 kilobases. And then the CT47 gene family, similar to gauge, ampliconic gene family spanning at least 25 kb. When we would run PacBio assemblies, for instance, on the X chromosome before, the assembly would often break at these regions because the reads were not long enough to span these sizes of repeat arrays. With the ultra-long nanopore data, we were able to assemble from end to end. One of the very neat consequences of that is now we can study the epigenetics and the structure of the entire chromosome, including the centromeres and the satellite arrays. And so this is a figure from our paper that was generated by Ariel Gershman at Hopkins, showing methylation patterns across some of these satellite arrays and across the pseudo-autosomal regions. If we look at the DXZ1 array highlighted here in middle and zoom in, this is showing the read depth of the ultra-long nanopore data across the array. And this is showing the predicted methylation status at all of the CPG sites as measured by the nanopore data and as called by Jared Simpson's nanopolish tool. And as you can see, the DXZ1 array is pretty heavily methylated across its length, but right in the middle of the array, there's a window of hypomethylation. And if we zoom further into that, we see the actual reads, and the CPG sites are colored blue if they're unmethylated, and they're colored red if they are methylated. And you see the very distinct pattern of hypomethylation right in the middle of the DXZ1 satellite array, as seen for the first time because we had never had an assembly to perform this type of analysis before. We've since repeated that analysis for multiple human chromosome centromeres within the CHM13 array, and we see the same pattern. For every centromere we see a region somewhat near the middle that has a hypomethylated signature. Another cool pattern within the DXZ4 array, where if you zoom in on the patterns here, you can see a periodicity to the methylation, where you have a periodic pattern of hyper and hypomethylation. Beautiful to see these patterns for the first time. And Ariel has some great follow-up analyses of this for both active and inactive X chromosomes, showing that the pattern of the DXZ4 array is different depending if you're an active or an inactive X. Most recently, we announced and released the completion of human chromosome 8, and this was work by Glenys Logsdon in Evan Eichler's lab. Similarly, here's the ideogram for chromosome 8 showing the regions that were gaps in the current reference. There was gaps in the telomere, the beta-defenstin gene cluster, the centromere, Q21 region, and the other telomere. We have now successfully closed and validated all of those gaps using a similar technique that we used for chromosome X, and repeated again some of those methylation experiments. And as I said, here you can see again a methylation analysis now for chromosome 8. And here, red is sites of methylation, blue is unmethylated sites, and you can see there's a region here towards the right side of the centromere 8 array, which has a pocket of hypomethylation. Glennis followed on and looked at SEN-PA attachments within that region and sees that there is an enrichment of SEN-PA that correlates perfectly with the hypomethylation. So now we're starting to get into functional analyses of human centromeres using this data. Further, Glennis had beautiful plots of centromere evolution, not just for this chromosome 8, but using our HiCanoe software, she assembled centromeres for other primate species and was able to show the evolution of those centromeres between the species and even the dynamics of the centromere satellite arrays within this human chromosome 8. Here, this dot plot is colored by sequence similarity, and you can see that this pocket 5 right in the middle of the array is most similar to itself, and then the divergence increases as you go towards the edges of the centromere, with the very edges of the satellite array showing the greatest sequence divergence, leading us to believe that they're the most ancient regions of this array. Definitely go check out this preprint from Glennis. It has a number of beautiful figures like this that help you understand the structure and evolution of human centromeres. So we were very happy to complete human chromosome X and human chromosome 8, but those were both very largely manual projects, X led by Karen and 8 led by Glennis. They took a year each worth of analysis and validation. And it was going to take us some time to repeat that for all human chromosomes. But within the past year, along comes a new technology called PacBio HiFi, or you might have heard it in the past referred as circular consensus sequencing. And this has some unique properties. Not only does it give you a relatively long read length of about 20 kb, but in comparison to the previous PacBio and Nanopore approaches, it also gives you extremely high accuracy, in some cases more accurate than Illumina sequencing, with 20 kb read lengths. And the way that this works is it goes through the standard PacBio template preparation where you take double-stranded DNA, you adapt these little barbells on the ends, the DNA polymerase takes that up and then incorporates bases, replicating that strand around and around the circle multiple times. The instrument then reads those as individual sub-reads and calls a consensus sequence on all of the passes that it makes on that circular molecule and gives you a consensus sequence for each individual molecule that it read, both forward and reverse strands. And that consensus sequence can very easily reach 99.9% accuracy, or a Q30 in a Phred score. When you then take those consensus reads and build an assembly out of them, you might have 30x depth of coverage out of those already high quality consensus reads. That makes it very easy to exceed Q50 or even Q60 consensus accuracy, which is 99.999 or 9999% accuracy. So this has some really great advantages. When you have that outstanding accuracy, it allows us to separate repeats, not just on the basis of their length, but by identifying the paralog specific variants within those repeats. So the variants, as I was showing you earlier on Karen's centromere figure, the variants that are unique to those individual copies, and using that information to separate out the repeats. The only con really is that the read length is limited to 20 kb. So initially it wasn't clear to us if these reads would be long enough, given their great accuracy, whether they would be long enough to resolve repeats in the human genome. But it was worth a shot, and we started playing with this last year and developing some approaches. And I'd like to tell you about the most recent approach that was developed by Sergey Nurk in my lab at NHGRI. And this is really contrary to what we were doing before, where we started with Nanopore and then polished with PacBio. In Sergey's approach, we're taking a graph first approach with the HiFi data. And the strategy is to build a very high quality and conservative assembly string graph, as it's called. And the first step to do is to clean up the reads. Usually genome assembly starts with a read correction phase. I said that the HiFi reads are extremely accurate. Their one Achilles heel is homopolymer sequences. And this has always been the case for Nanopore and PacBio, that when you have a homopolymer here like cat, I'm not sure if that's 4 or 5 A's. And so the first thing we do when we build a HiFi string graph is we compress all of those homopolymers down so that cat becomes cat with 1A. And we do that for every sequencing read in the input. And we consider only those homopolymer compressed reads when we're building the graph and computing overlaps and so forth. We then do some read cleaning to get rid of any chimeric products or missed adapters. And we also do some very conservative corrections. So if I see 20 HiFi reads piled up in a row and only one of them differs from the other 19, it's very likely that one difference is an error. So we correct that error to match the consensus. And from those corrected homopolymer compressed reads, we then build a string graph only from perfect overlaps, meaning when we're overlapping 2 sequences before, like we were talking about before with the gel reads, we're only going to allow ourselves to overlap and connect those reads if they have 0 differences in the homopolymer compressed space. So what I'm calling perfect overlaps. And we're also doing that for very long overlaps of say 7,000 bases. When you do that, you're able to construct a graph that surprisingly was very complete and had very little ambiguity across the whole genome. But we were left with a number of tangles, which is the very technical term for when the graph isn't just a linear path. And I'll talk about those in a bit. But just a note here, when those tangles are easy, you can actually formulate it as a Hamiltonian walk problem and say, if I want to visit every node in this graph once, where each of the nodes represents a bit of unique genomic sequence, in many cases, there's only a single walk through that tangle that preserves this condition that we want to visit each node once. We can also come in with the ultra-long nanopore data and align those reads to the graph to help guide a path through these tangles. After we resolve those tangles, we go back to the HiFi data, we decompress the homopolymers, and we call a consensus sequence, which helps us improve the accuracy in these homopolymers or simple sequence regions. And at the very end, we output only the HiFi consensus. We're using only the nanopore data for detangling, not for consensus calling in this case. And so Sergey was just playing around in the spring really to understand the HiFi data and to understand the graph construction process. And almost as a side effect, generated some absolutely beautiful assembly graphs for CHM13. And when we saw these graphs for the first time, it really excited us by the prospect that we could finish the human genome using the HiFi data and using these graphs. And the 2 primary observations were that, as I'm showing here, this is the whole genome string graph for CHM13 as output by Sergey's process. Nearly all of the chromosomes, with the exception of the acrocentric chromosomes down here in the bottom right, all of the other chromosomes are output by this process as a single connected component, essentially from telomere to telomere. In some cases, a single chromosome might have 2 or 3 components. But to our surprise, none of these chromosomes were interacting with each other in the graph, meaning there was no repeats in the human genome that were so similar between chromosomes that it confused the assembly process. And every chromosome was very distinguishable on its own. The second observation was that these individual chromosomes were mostly linear reconstructions. When I zoom in, I'll show you in a second, there are some local structures, but when you zoom out at a gross scale, it's very clear what chromosome 1 is and what chromosome 2 is, and it's largely linear. And this was really shocking to us. We did not expect the genome to look this way. We knew that there was segmental duplications and satellite sequences shared between human chromosomes, but apparently they've diverged enough in terms of evolutionary time that they can be separated on the basis of HiFi data alone when you're considering a perfect string graph with 7 kb overlaps, as we're doing here. So given this great preliminary data, we decided to hold a summer finishing workshop where we were going to dig into this assembly graph and see how many chromosomes of CHM13 we would be able to finish with the help of the world's experts in genome assembly and satellite DNAs and so forth. And so we convened in June of 2020. Despite the invading Roman armies at our doorstep, we focused on these assembly graphs all summer, almost as a way to divert our attention from the other perils of the world. And we split up into working groups on genome assembly, the satellite DNAs, the RNA gene arrays, segdupes, validation teams, annotation teams, curation teams, and we went at it. And here's what we were looking at. Here's 3 chromosomes that we categorized as good on the basis of the assembly graphs. And this is the entirety of chromosome 10, 20, and 8 as they looked in our initial HiFi string graphs. And I'll draw your attention to one type of motif where you have what I would call a simple bubble, where you have a long string here, which is representing a string of DNA, and then there's branches, 2 strings in the bubble, back to a single unambiguous string. This is a heterozygous site. I said earlier that we picked CHM13 because it's mostly homozygous. That's the rule, but there are exceptions. It is a diploid genome. There are a few thousand sites of heterozygosity, and this would be an example of one of them, where the 2 chromosomes are identical in this region. There's a heterozygous variant that splits the graph into a bubble because we're requiring all of these sequences to be perfect. So even a single SNP would cause this type of bubble. Back again to a continuous piece of DNA. Just to correct myself, I said a single SNP. We had automated graph processing that would go through the graph, and any of these heterozygous differences that were less than, say, about 2 kilobases, we would automatically collapse into a single string. Where they're broken out here are the largest, more structural heterozygous differences. The second type of motif on this slide is a bit of a more complex tangle, but this is still what we would call an easy tangle, where in these cases, there's generally a single path through these tangles that visits each of the nodes. And Sergey further developed a method that allowed us to label each of these nodes with depth of coverage. So in the case where you might have to go through a node twice, we're able to tell that based on double the expected depth of coverage for that node. Here's an example of some bad chromosomes. Chromosome 2 and 18 presented a little bit more difficult of a challenge. You can see much more complex graph structure here for chromosome 2. And 8 kind of ends in this little lollipop structure, 18, as it approaches the telomere. These presented a greater challenge, but with some sleuthing and the addition of Nanopore data, we were able to resolve these chromosome graphs as well. But the ugly is really the acrocentric chromosomes in this case, which I mentioned before. And these were the only human chromosomes that were brought together into the graph as a single tangle. And here I'm showing you a junction of the graph where you have 4 different human chromosomes all linked together by sequence similarity. And this is in the rDNA regions of the acrocentrics. So as background, the human acrocentric chromosomes are the 5 acrocentric chromosomes. The P arm of each of these chromosomes contains the rDNA repeat unit, which contains the rRNA genes for the RNA components of the human ribosome. Those are labeled here in pink. And you can see that there's a little bit of chromosome towards the telomeric side. Then you have the rDNA array, and then you go back into the chromosome before you hit the centromere. And so that's the structure we see in the graph here. I've labeled the telomeric ends of the graph. Those are pretty easy to resolve with the HiFi string graph. Then you come into a central tangle here, which is directly adjacent to the 4 rDNA clusters, which are these really massive tangles in the graph because the rDNA unit is about a 45 kilobase exact repeat that's in 300 or 400 copies in the genome. It shows up in the graph as these very fragmented clusters. And then you extend out of those clusters to the centromere of these 4 chromosomes. There's actually a lot that you can gather, even though this is complicated. There's some key clues into the structure of the rDNA regions just looking at this graph. 2 of the most important clues are that the rDNA clusters are separated in this graph and appear to be chromosome specific. It's been previously assumed that these rDNA clusters undergo frequent recombination with each other, kind of homogenize themselves across the chromosomes. But in this graph and in this cell line CHM13, they appear to assemble independently, suggesting that there's chromosome specific variation within them. Also upstream of those regions and what's called the distal junction, there's evidence of very recent recombination of the genome at that location. Even given those complexities, with the help of Nanopore, we were able to thread our way through this graph, aligning Nanopore reads to these individual sequence nodes and then guiding a path through this tangle, successfully resolving all 5 of the acrocentrics with the exception of these rDNA clusters, which are a real mess and we're still working on. But we now have assemblies of all 5 of the acrocentrics from telomere to telomere, with the only gap being a sequence of ends where we haven't quite resolved these clusters yet. So to make a long story short, this project over the summer was hugely successful. And on the basis of these HiFi assembly graphs, we were actually able to complete the first sequence of a human genome, with the star exception being the 5 rDNA clusters. In comparison to GRCH38, the T2T CHM13 genome shown here, we have 23 chromosomes assembled with no Y. We have 0 unlocalized or unplaced sequence, meaning we know where every sequence that was coming off the sequencer goes in the assembly. There's no leftover bits. We have about 3 gigabases of assembled sequence, so that's adding around 100 megabases of new ACGTs compared to GRCH38. We're removing about 150 megabases of gaps, so ends in the current reference versus ends in the new reference. And key, the ends that are in the new reference are known gaps. We know that these are the rDNA arrays. We know that those are just tandem duplications of a highly conserved sequence. And so there's no missing sequence from our assembly. It's all there. We have graphs of the rDNA arrays, and we're working on understanding the variation within them at this time. And then finally, the quality of this assembly is very, very high. The current reference is built on a mosaic of multiple different human haplotypes, and so it's very hard to judge the quality because we don't really have the original material anymore. For this, we have the cell line, and we can use that for various validation assays. And we currently estimate maybe Q60 or Q70 consensus quality. We have no known homozygous structural variants. There's no inversions as measured by strand-seq. This is a very high quality assembly. We're continuing to validate it, but we have high confidence that it's correct and accurate. We estimate the size of this human genome is 3.057 gigabase pairs, and we're adding about 100 to 190 megabases of new sequence compared to GRCH38, and that's about 3 to 6% of the human genome length. I don't know the exact number because it depends on how you do the alignment. The lower bound is this 100 megabases of new bases that we're adding. The upper bound is alignment based on this Newtoner command down here. And in the last couple of minutes I have, I'm just going to take you on a quick tour via dot plot of what some of these regions look like. This is the centromere and the HSAT2 array on chromosome 1. This is spanning maybe 30 megabases of sequence. Oh, I have a scale bar here, 20 megabases of sequence. And the centromere in chromosome 1 shows this very unique inversion pattern where the forward matches are in purple, the reverse matches are in blue. And you'll see a lot of these satellite arrays have a checkerboard pattern, meaning there's large inversion blocks buried within the arrays. And then this big HSAT2 region on chromosome 1, which I really didn't think we would ever get across. It's over 10 megabases of human satellite. We've assembled it with the help of HiFi. Showing now the junction between that HSAT array and the rest of the Q arm. This is the T2T assembly versus GRCH38. Again, showing a lot of inversions in the reference versus our assembly. Now, these regions are known to be prone to inversion in the human population, but we are finding that our assembly looks a lot more correct in regions of inversions than the current reference. And although these are polymorphic in the population, we're confident that our assembly of CHM13 is an accurate reconstruction of a haplotype, and we're not hallucinating inversions or sequences. Lastly, here's a very cool region. This is the full region of the P arm of chromosome 14 assembled, again shown in dot plots here. The little gray blocks is the rDNA gap. Everything else is assembled sequence, showing great patterns of segmental duplication, inversions. This is a large palindrome towards the centromere. These are HSAT arrays in the middle. If we zoom in again and show a dot plot blown up, we have forward and reverse matches in that checkerboard pattern within the HSAT arrays here, buried within a bunch of segmentally duplicated arrays. Really cool structure. So finally, how do we know it's right? Well, my strategy is to release early and release often. So we put this assembly out immediately, and we're going to let the community decide if there's any issues. This worked out well in the past, and Evan Eichler's lab has since joined the consortium and really improved our assembly of the X chromosome. There's the Corin test, as I call it, which is coined after Sergei Corin in my lab, which is if I synthesize this assembly from scratch, would it make a viable cell? The answer for GRCH38 is obviously no, because there's so many gaps. But maybe for CHM13, we now have all of the sequence in the right location and the right copy number, if you were able to synthesize a whole human genome, this might produce a viable cell. What we actually did is what I would call a likelihood test, and we're looking to see if the sequencing data matches our expectation, that we don't have unassembled reads, there's uniform depth of coverage, the only errors that are left are random sequencing errors, and there's no inversions or fusions as measured by strand-seq and HiFi. So to do that, we mapped all of the raw data back to the assembly, and we ran variant calling analyses using Tandem-Quast, deep variant, and sniffles. Here's an example showing what a heterozygous variant looks like, where some of the reads show an insertion and some show a match to the reference allele. Those were what we were called false positives. As long as we're matching one of the alleles, we're happy. In the end, we had to make very few corrections to this assembly. We added 4 kb of telomere to chromosome 18. We fixed 1 deletion to insertion structural variants, and we only made about 1,000 small variant corrections, which confirms our initial prediction that this assembly exceeds Q60 in terms of quality. Here's an example before and after correction of one of those. This is an initial false insertion in the assembly where all of the reads from variant calling call a deletion. After the correction of that, everything looks good. So I'm happy to announce that this assembly is available and released to the community. You can download it from this link, genominformatics.github.io. There's a short announcement with a preprint in progress. We're releasing both the unpolished and the polished assembly. And coming soon is a preprint on this assembly and analysis. We're going to make publicly available a genome browser annotation. We'll submit this assembly to GenBank. And we're still working hard on the rDNA array and chromosome Y. And I expect we'll have results for those in the coming months. And just to emphasize, we only finished this assembly about a week ago, and we encourage everybody to have a look at it. And please reach out to the consortium if you'd like to contribute. Release early, release often. So I'll just leave you with summary thoughts that HiFi is a very powerful new data type that allows you to generate extremely accurate yet continuous assembly graphs. Nanopore data is still useful as a complementary technology for resolving tangles and filling any gaps. The human genome is finally complete. We can now look at what we've been missing in that 5 to 10% over the last 2 decades, and we can start building human pangenomes, extending from just a human reference to a pangenome reference that encompasses all human genetic variation. And that's essentially where the T to T is headed next. We want to automate these assembly methods. We're going to test those automated methods first on a panel of haploid X chromosomes taken from male cell lines. And we're going to eventually push into doing this for diploid genomes so that we can now have a diploid complete T to T assembly for any genome at any time. And with that, I would just like to thank the fabulous consortium that we've built up over the past couple of years, our industry partners as well, listed here on the right, my co-chairs of T to T, Karen Mika and Evan Eichler, and Sergey Nurk, who led the assembly working group over the summer and developed these methods that were key to assembling the first complete T to T human genome. If you'd like to join this effort, please contact us for more info. And with that, thank you for your attention.
Okay, well, thank you very much, Adam. That's a fabulous talk. We all applaud. I don't know how we do that. I'm going to applaud in my screen.
Thank you, Richard. It's good to be with you live. Thank you much for your kind introduction earlier.
Okay, so there's questions already arriving, and maybe I'll start with a question from Rob Waterhouse. So some of them are anonymous and some of them have names. He says, great quality, clearly impressive amount of work. Is this overfitted for kind of ultra important genomes, or is it flexible enough to work on others?
Yeah, hello, Rob. Good question. It is very much overfitted for the human genome, particularly because we're dealing with this mostly homozygous CHM13 line. So if you have a homozygous genome or a haploid genome, the approaches that we used for CHM13 would work quite well. And we will probably extend this work to maybe finish the genome of a doubled haploid zebrafish or something like that, for instance. But if you're talking about diploid genomes, our current approaches are suboptimal for that. And there's still a lot of methods development that's needed in the coming year or two to get T to T diploid genomes. But that's something the assembly groups are working very hard on.
Okay, so here's a question about the methylation results from an anonymous questioner. Is there is there active hypomethylation at the centromere, or is it just possibly that the methyltransferases can't access the DNA in that region?
Yeah, that's the old chicken and the egg problem of epigenetics, which came first. It's really a question for Karen Miga and the satellite community who are digging into this function. But that's where they're coming into the ChIP-seq and the cut and run experiments to actually measure the attachments of the proteins at these regions and try to understand where the active centromere is. And I showed a little bit of a result early on from Glennis's work where she had mapped some ChIP-seq data showing the SEMP attachments at the centromere.
Okay, here's a question about validation. You talked about going through lots of different validation, and the question is, actually, I've just lost it. Would it be useful confirmation to map back-end sequencing and restriction enzyme maps that were originally done for the HGP? I guess the answer is you want to look at this particular line, but maybe you could tell us, what is the best independent data for validation?
Yeah, it's a hard question. The answer to the first question is no, that wouldn't be very useful because they're coming from different individuals. And so you wouldn't be able to tell what's a true structural difference versus an error. The second answer is that we do actually have a pretty well characterized back library for CHM13. And a number of those backs have been sequenced and finished by the Eichler lab. In digging into those, the differences that we now find between our assembly and the back library, when you chase those differences down, there are errors in the backs and the back assembly rather than our whole genome assembly. Full validation is a tricky question, especially in the centromeres, but we're throwing whatever we can at the problem, and we have some ddPCR estimates for array lengths. We're doing a number of southern blots on restriction digest to show that the digested fragments match our prediction. But really, it comes down to mapping all of this complementary data, Nanopore, optical map data, HiFi, and show that they're in agreement and there's no obvious discrepancies between the raw data and our assembled output.
Okay, maybe we can have a couple more questions. So there's a bit of a gap. I was going to ask, so this is from Yvain. How can you be sure that one difference in 20 fragments is not a SNP? That your error correction didn't erase things which actually are biological? Yeah,
and that brings up the good issue of variation in the population of cells where there's low-level variants that are really there in some subpopulation of the cells. And this does exist. We do see that when we look hard enough, as opposed to the 50-50 coverage we would see for a heterozygous variant. In those cases, we're happy to take the majority allele. And so the correction strategy is as conservative as we can make it, and it's really only one difference that's willing to be corrected at this point. In the cases where there's a true heterozygous allele, where there's a 50-50 split, as I said, there's a few thousand of these in this cell line that we've identified. We have to pick one allele. In most cases, we pick the larger of the two alleles. In the cases where there's a majority variant, maybe 90% coverage versus 10%, we take the majority variant. But we are trying to catalog and characterize that variation as we go along.
Okay, maybe I'll ask, there's a more general question as the final question. Cool work, Adam. I want to know, what are your thoughts about the general acceptance, the statement that the 2000s human genome was complete?
Well, I hope I
people will
that it's not.
And do you think people will look back now, at what you've done, and you've claimed to have completed it, and they'll say, "Oh no, he didn't complete it."
Oh yeah. So can we officially claim that it's completed now? Well, that's why I was careful to put an asterisk on the slide that claimed we had completed it, because we still have the rDNAs to work out, and we know that there's variation within the individual copies that we need to pin down. So I won't say that we've finished it off yet. We still want to complete the Y chromosome. But I really view this project as a technology development project, that we're really pushing the limits of the sequencing technology and the algorithms. And the real hope is to make every genome a complete T2T genome in the future. And so we'll look back and not have a single reference genome, but we'll have enabled the technology that lets every genome be a reference genome going forward.
Okay, well, thank you again from me, but also I think there's multiple people who've supported what Marcella has said, and I'm sure that speaks for everybody else. What an amazing talk, Adam. So thanks very much.
Great. Well, thank you very much for having me, Richard.
Okay, and now there's a 5-minute break before the next set of sessions start.